Hello, everyone, and thanks for joining today. I would like to especially thank the organizers for inviting me. Today, I'm going to talk about a novel technology, the seeding amplification assay as a novel tool for the diagnosis and stratification of patients with multiple system atrophy. I have no conflict of interest, and this is the outline of my talk, where I'm going to try to answer to these 8 questions. The first one, and the one most of you already know the answer, is what is MSA? So multiple system atrophy, or MSA, is an adult onset progressive neurodegenerative disorder. MSA is regarded as the most aggressive synucleinopathy, as it is characterized by rapid progression leading to severe disability within 5 to 6 years and typically death within 10 years of symptom onset. But why do we say that MSA is a synucleinopathy? This is because recent molecular studies have identified abnormal deposition of proteins as the neuropathological hallmarks of neurodegenerative diseases. Currently, the pathological classification of these diseases is based on the predominant proteinopathy. The presence of the 140 amino acid protein alpha-synuclein as a major component of MSA inclusions linked MSA with Lewy body disorders such as Parkinson's disease and dementia with Lewy bodies. Collectively, these disorders are now termed synucleinopathies. Lewy body disorders are classically distinguished from MSA by distinct cellular pathology. Although both conditions accumulate alpha-synuclein in a variety of cell types, MSA is characterized by oligodendrocytic inclusions, called UCIs, as you can see here, while neuronal alpha-synuclein pathology, as you can see here, predominates in Lewy body disorders. Because of the classification of MSA among neurodegenerative synucleinopathies, the role of alpha-synuclein is falling, and aggregation has been a major focus of research and will be the focus of this talk. However, I want to also point that in addition to alpha-synuclein misfolding, altered neuronal iron homeostasis, mitochondrial dysfunction, failure of myelination and trophic support, and neuroinflammation may also have very important roles in MSA. But what causes the symptoms? The defining clinical symptoms and signs of MSA are related to dysfunction and pathology in the striatonigral, olivopontocerebellar, and central autonomic systems. Core features arising from dysfunction in these domains are Parkinsonism that responds poorly to levodopa, cerebellar ataxia, and autonomic failure, which occur in different sequences and combinations, and together with additional features, lead to severe disability as the disease evolves. Then, how can we diagnose MSA? A group of experts published diagnostic consensus criteria in 1998, and a revised version from 2008 subsequently became the clinical diagnostic gold standard for MSA. However, diagnostic performance of these revised criteria has remained suboptimal, with low sensitivities for possible and probable MSA at the first visit, and more than 20% error rates even at the last visit in clinical pathological studies. Revised criteria for clinically established and clinically probable MSA were developed by a working group commissioned by the International Parkinson and Movement Disorder Society, MDS, and published in 2022. These criteria include now mandatory use of magnetic resonance imaging biomarkers for diagnosis of clinically established disease, and also list additional diagnostic biomarkers that you can check in this paper over here if you are interested. Also, and for the first time, these criteria include a category of possible prodromal MSA for use in research studies. But why do we need novel biomarkers in MSA, and why this prodromal phase of MSA is important? Well, similar to other neurodegenerative diseases, such as Alzheimer's disease or Parkinson's disease, the onset of molecular pathology underlying MSA antedates its clinical diagnosis by several years. Although the onset of the biological disease may be clinically silent, prospective studies have provided evidence for a prodromal phase of MSA. Sensitive and specific biomarkers are needed to enhance early diagnosis, and hopefully with the new disease-modifying therapies that are now being tested in clinical trials, diagnostic accuracy in MSA is key, as well as having the tools that enable us to detect target engagement and progression effects of this disease-modifying intervention. In this very elegant review published a couple of months ago, there is a summary of all the new assays, biological fluids, and tissues that are being used as a possible novel biomarker for multiple system atrophy. In this talk, I'm going to focus on the use of seeding amplification assays, but I encourage you to read this article if you want to learn more about other assays and proteins that are also being tested to get in the future a better and more reliable MSA diagnosis. Then what are seeding amplification assays? So seeding amplification assays, also known as RT-QuIC or PMCA, were originally developed for the diagnosis of prion diseases, where they revolutionized the way these diseases were diagnosed, achieving almost 100% sensitivity and specificity. The basic concept underlying the RT-QuIC or seeding amplification assay is that a protein aggregate or a bad protein is able to misfold a protein monomer. RT-QuIC involves shaking a specimen, harboring a small amount of disease-associated protein with monomeric proteins in multi-well plates. Based on the concept of seeding, proteins capable of misfolding will convert the monomers into aggregates with monomeric recombinant proteins in multi-well plates. And the real-time detection of these fluorescence permits the measurement of kinetic differences between the seeding properties of different samples. If the sample added to the assay does not have bad synuclein, let's say, there will be no fluorescence, as you can see in this curve over here. Whereas if the sample added has bad synuclein, it will generate fluorescence, as you can see in this image. The great advantage of this method is that it can detect misfolded protein in bodily fluid samples, such as CSF or brain homogenates. Furthermore, recent studies, as you can see here, have shown that in addition to the detection of prion protein, alpha-synuclein and different isoforms of tau protein can be reliably and sensitively detected by this method. In this example, you can see that the RT-QuIC technology or the seeding amplification assay technology has been successfully adapted to detect alpha-synuclein in the cerebrospinal fluid or CSF with remarkable sensitivity and specificity, more than 90%, in the differentiation of Parkinson's disease patients from healthy individuals. However, despite the reliability of these assays to consistently detect alpha-synuclein seeds in Lewy body disorders, attempts to detect alpha-synuclein from multiple system atrophy patients have proven challenging using this assay, which was quite surprising because, as I mentioned in the introduction, MSA is considered to be the most aggressive synucleinopathy. But as this is a very active field in the neuroscience now, a couple of months after that paper I mentioned before came out, this paper was published in Nature by Claudio Soto's group. In this paper, using a slightly different assay, they were able to obtain MSA positive curves, as you can see here, and they could even differentiate the MSA from the PD patients using the THT fluorescent profile, as you can see here. This is the Parkinson's disease curve, and this is the MSA curve. So we were wondering what could be the difference between these two methods. And we had the theory that these divergent findings are likely the result of different conditions in the reaction buffers used in these studies, and that using suboptimal conditions, the absence of seeding activity could reflect the absence of seeds, but also reflect an inability of the specific RT-QuIC conditions to support the amplification of those particular seeds. So one thing that we did in the lab was we wanted to identify the optimal assay conditions that favor MSA seeding activity by systematically modulating the ionic and cationic composition and the pH of the alpha-synuclein RT-QuIC reaction buffer that you can see if you are interested in this article here. So the effect of ionic composition and pH and alpha-synuclein aggregation has been widely studied, but not in the context of systematic comparison of a spectrum of reaction buffers seeded with brain homogenates from MSA and Parkinson's disease. To do this experiment, a region rich in Lewy body pathology, the substantia nigra from a PD patient, and a region enriched with GCIs, these oligodendrocytic inclusions, typical of MSA in the cerebral white matter from an MSA patient, were carefully microdissected using a 4mm brain tissue punch and subsequently processed into crude homogenates using PBS. In these two cases, conventional immunohistochemistry for disease-associated alpha-synuclein demonstrated abundant Lewy bodies in the substantia nigra of the Parkinson's disease patient and GCIs in the white matter of the cerebellum of the MSA patients. Next, and using a combination of biochemical assays, as you can see here, we confirmed that the synuclein found in the brain of PD and MSA patients was structurally different. Following the characterization of these brain homogenates, to identify the optimal assay condition that favor MSA seeding activity, we seeded 168 different R-to-quick reaction buffers using 5 micrograms of MSA-derived or PD-derived PBS soluble fraction. We then quantified 5 parameters, as you can see here in this image, and we calculated the fold separation of these parameters examined by dividing the values obtained by the MSA and the PD brain homogenates, and then we generated heat maps. This is the heat map, and the red color of the cell indicate that the buffer is more favorable to detect PD synuclein, whereas the blue color indicate that the buffer is more prone to detect alpha synuclein. For example, all these buffers highlighted in yellow are more prone to detect MSA synuclein, and these buffers here that are in red are more able to detect Parkinson's disease derived synuclein. After the analysis, our analysis revealed that 19 out of 168 buffers were able to detect predominantly PD-derived alpha-synuclein aggregation, showing at least twofold difference between PD and MSA curves, while 40 out of 168 buffers showed an at least twofold difference between MSA-derived alpha-synuclein and PD-derived alpha-synuclein aggregation. From these 40 buffers, the 10 conditions that display the highest differences, highlighted in yellow here, you can see here, were selected to further examine the parameters of kinetic curves. Based on the raw values obtained with each condition, we selected 2 winning buffers over here. The first buffer was 50 millimolar glycine pH4, and the second buffer was a 40 millimolar phosphate buffer pH 8. Once we had the 2 optimal buffers selected, we validated them in a larger and multicentric cohort of disease samples for their performance in detecting MSA-derived alpha-synuclein seeding activity and differentiated it from PD-derived alpha-synuclein seeding activity. To avoid also any regional difference, we microdissected the substantial nigra of 15 subjects with MSA, 15 subjects with PD, 5 subjects with progressive supranuclear palsy, and 5 controls, and processed the samples into PBS-soluble homogenates. All these subjects have a detailed neuropathological examination. In panel A, and to illustrate the typical profile of alpha-synuclein R-to-quick aggregation from samples of PD and MSA, we plotted data from one representative MSA, as you can see in blue, one representative PD, as you can see in red, and one control, as you can see in green, amplified under the buffer 1. As you can see in panel B and C, the maximum fluorescence and the area under the curve were consistently different between PD and MSA, with samples from patients with MSA aggregating faster, while always reaching a lower fluorescence plateau than samples from patients with PD. In the panel E here, we observed that when we amplify under buffer 2 condition, the same cases, samples from patients with MSA, still aggregated faster, as you can see here, but in contrast, they reach a higher fluorescent plateau than those from patients with PD. Furthermore, significant differences in the maximum fluorescence and the area under the curve were observed between MSA and PD patients, as you can see in these panels here. But now that we have, or that we achieved a specific buffer for MSA, how can we use this assay for the molecular classification of MSA? One thing that we found when we were evaluating the R-to-quick data generated in the validation phase is that we observed up to 10-fold differences in the area under the curve and the THT max values among the 15 MSA subjects included in our study. To test the hypothesis that heterogeneity in alpha-synuclein seeding exists in MSA, we selected 6 from the 15 cases or MSA subjects and classified them as low seeders, intermediate seeders, and high seeders according to their alpha-synuclein seeding behavior. Once we knew that the different patients had different seeding capacities, we also wanted to evaluate whether there were also seeding differences within the brain of the same patients. We evaluated these 13 different brain regions, and from each case, as you can see in this image, we microdissected and subsequently prepared protein homogenates from the regions that you can see here. All the samples were run in quadruplicate, and the R-to-quick curve obtained by each reaction was evaluated as a measure of alpha-synuclein seeding behavior. The area under the curve were converted into scores ranging from 0 to 3. And as you can see here, with these values, we generated a heat map of alpha-synuclein seeding to illustrate the difference between brain regions and patients. Interestingly, based on the mean of the area under the curve values across the brain regions, the two patients that we classify as high CIDR had significantly higher values than the intermediate or the low CIDR patients. When the different brain regions were examined individually, the Pons base showed the highest alpha-synuclein seeding. Surprisingly, alpha-synuclein extracted from two of the most affected regions in MSA, the cerebellum and the putamen, exhibited the lowest seeding. A possible explanation is that the analysis of the alpha-synuclein seeding was performed using what we call the PBS soluble fraction. It is possible that the seeding activity of alpha-synuclein could diminish over time as the more soluble seeding competent alpha-synuclein species present at early stages of the disease become sequestered in larger and insoluble aggregates at later stages of the disease. This could then result in reduced seeding of alpha-synuclein extracted from regions affected earlier in the disease course. Finally, we aimed to evaluate whether the heterogeneity of alpha-synuclein seeding was reflected in the immunohistochemical findings in the brains of the same subjects. Thus, we performed a semi-quantitative analysis of the alpha-synuclein aggregation burden using an alpha-synuclein conformational antibody called the 5G4. In this analysis, the cerebellum and the putamen show the highest aggregated alpha-synuclein, as you can see here, here, and here. And when we compared back-to-back the heat maps generated from the alpha-synuclein seeding capacity and the ones generated from the histological semi-quantification of aggregated alpha-synuclein burden, we realized that both examinations are complementary as they measure different stages of alpha-synuclein misfolding, but that performing RT-quick assays on top of conventional histological analysis show more heterogeneity between different brain regions of a given individual and between individuals also, that goes beyond immunohistochemical observations and can pave the way for future subclassification of MSA. Finally, how can we use them or these assays for the molecular diagnosis of MSA? To show you an example of how this technology can be used for the molecular diagnosis of MSA, I would like to quickly go over a nice study that we published last year, where we took advantage of the method that I showed you to overcome one of the major challenges in achieving early differentiation of neurodegenerative Parkinsonism, the inconsistent detection of disease-associated alpha-synuclein in multiple system atrophy patients. So our idea was to validate the assay for CSF, or cerebrospinal fluid. However, one caveat to the widespread application of RT-quick assays is the reliance on the use of invasively obtained CSF to catalyze the reaction, which may pose a challenge for feasibility of large-scale longitudinal studies and also for implementation in the clinical practice in the hospitals. So as there are already several reports using that RT-quick or showing that RT-quick has been successfully performed using skin biopsies from patients with Creutzfeldt-Jakob disease, a prion disease. And also there are a couple of reports showing that this can be used also to distinguish PD from healthy controls. We optimize our novel assays to assess alpha-synuclein seeding activity using a small single 4mm cervical skin biopsy from clinically diagnosed patients with MSA, Parkinson's disease, progressive supranuclear palsy, and healthy controls. As you can see here, cutaneous alpha-synuclein RT-quick was positive in 9 out of 10 patients with MSA, 11 out of 13 patients with Parkinson's disease, and only in 3 out of 20 healthy controls and 1 out of 7 patients with PSP, achieving a combined sensitivity of 87%, a sensitivity of 87% and a specificity of 85% for differentiating synucleinopathies versus non-synucleinopathies. However, and interestingly, the assay was not able to clearly distinguish MSA from PD based on the seeding kinetic alone, as it occurred, if you remember, when we used brain homogeneics. Thus, we needed to go further, and based on the published success of circulating neurofilament light change as a potential early marker to distinguish patients with Parkinson's disease from those with atypical Parkinsonian disorders, we analyzed the neurofilament light change concentration in all subjects using an ultrasensitive detection system called single molecular array, or SIMOA. As expected, serum neurofilament light change was significantly higher in patients with MSA than in patients with PD, as you can see in the graph here, with no difference in circulating NFL found between PD patients or controls and between MSA and PSP patients. So we proposed in this article a two-step diagnostic approach combining skin RT-QuIC and measurement of circulating NFL assay, where we could differentiate PD patients from those with MSA and PSP with 100% sensitivity and specificity, and MSA patients from those with PD with 100% sensitivity and 93.3% specificity. These promising results here, of course, required further exploration before our approach can be widely applied, but this approach has the potential to ensure that trials for therapeutics targeting alpha-synuclein recruit individuals with a positive cutaneous alpha-synuclein RT-QuIC and distinguish MSA from PD as necessary. Finally, I would like to show you two very recent articles that will potentially change the way we diagnose neurodegenerative diseases without the need of a lumbar puncture or even without the need of a skin biopsy. The first paper that I want to show you was published earlier this year in Nature Medicine by Japanese colleagues led by Dr. Hattori. In this paper, they describe a new method where they were able to isolate aggregated forms of alpha-synuclein present in the blood of patients with synucleinopathies. Interestingly, they were able to detect aggregated forms of alpha-synuclein in the blood of 95% of patients with Parkinson's disease and in 90% of patients with Lewy body dementia. But when they examined the blood of patients with MSA, as you can see here, 39 patients with MSA, they only find aggregated synuclein in 64% of these patients. Interestingly, and despite the fact that they did not find aggregated synuclein in all the MSA patients included, when they examine and compare the photocopies of the aggregates derived from the MSA and the PD patients after they performed the seeding amplification assay, the authors found that these fibrils were different between MSA and PD under the electron microscope, a technique that enabled us to look at the structure of these aggregates with this detail. As you can see here, this is the fibril of a PD patient, and this is the synuclein fibril of an MSA patient. And if you see here the 3D structure, you see that the Lewy body disease fibrils and the MSA fibrils are very different. And also, when they treated some cells, this is a cell culture, they treated cells with some of these fibrils, they added the fibrils to these cells. The author also realized that the aggregates formed in these cells were different between the cells treated with MSA fibrils and those treated with PD fibrils. This, as you can see here, these are PD cells seeded with PD-derived fibrils, and these are cells seeded with MSA-derived fibrils. The green dots are aggregates, and as you see here, the MSA-derived fibrils produce PC and DC aggregates that you can see here, DC and PC, and the PD produces mainly FL, what the authors call FL aggregates. And if you see here, basically the morphology of these aggregates are different. Of course, these results suggest that the synuclein found in the blood of these patients resemble of the one present in the brain, and this will need to be validated by the other groups. But this study may be the first step towards a blood test to diagnose synucleinopathy. And finally, this last piece of work that I would like to show you today also shows the effort from our German colleagues to also establish a blood test for synucleinopathies using seeding amplification assays. In this case, instead of capturing the circulating aggregates of synuclein in the blood of Parkinson's disease patients or MSA patients with an antibody, they isolated the circulating exosomes from the blood. So what are exosomes? So exosomes are small vesicles secreted by cells into the extracellular space. These vesicles can transport lipids, nucleic acids, DNA or RNA, and proteins between cells, and also they can carry, now we know, that they can also carry synuclein aggregates. This, together with the fact that these exosomes can cross the blood-brain barrier, and the ability that we have now, as you can see here, to recognize the origin of these exosomes using antibodies that are specific of the cell membrane. So now using an antibody, we know that this exosome was secreted by a neuron, or this exosome was secreted by an oligodendrocyte, or that this exosome was secreted by an oligodendrocyte. We can isolate, or what this German group did was like isolate the exosomes derived from neurons from patients with PD and compare them with patients from the exosomes derived from control individuals. They extracted the protein of this exosome, these neuronal derived exosomes, and they performed RT-QuIC. What they discovered is that the exosomes derived from patients with PD were positive for RT-QuIC, meaning that these exosomes contained misfolded alpha-synuclein, pathological alpha-synuclein, and that the exosomes, the neuronal exosomes derived from healthy individuals, they were negative, meaning that these exosomes didn't carry any alpha-synuclein or any aggregate of alpha-synuclein. Unfortunately, this study did not examine MSA patients, but thanks to the generosity of the MSA coalition, our group is now studying whether blood exosomes of MSA patients can be used as a diagnostic tool also for MSA. To finalize my talk, I would like to highlight some of the take-home messages. Novel biomarkers are vitally needed to diagnose MSA with high specificity and sensitivity in the early stages of the disease. Seeding amplification assays, once validated, could be a rapid and affordable assay that could be used for this purpose. Seeding amplification assays will also be useful tools for the molecular classification of MSA, which will allow us for the development of new personalized therapeutic strategies for this devastating disease. Thank you very much. And I would like to sincerely thank all the persons that have helped me in these projects, especially my lab colleagues here, and to the funding sources that have made these projects possible. Thank you very much for your attention. And if you have any doubts, please reach out to me. Thank you.